so we're going to start the first part of the last chapter, which is about nucleic acids. So remember, we made the changes to the schedule that we're not going to end on chapter 16. We're going to end on chapter 15 because you will talk about metabolism in both nutrition and A&P. All right, so nucleic acids, our last big group. So we're going to start off with some definition type stuff, and you'll see why we started with this when we get to the end, because we're going to talk about DNA. So a chromosome is an elongated thread-like structure composed of protein, so everything we talked about in chapter 14, and DNA, stuff we're going to talk about today and on Friday. Sorry, I'm recording this. Today is 9/28 in my world, so still kind of trying to figure out where we are in a few weeks. So that contains the genetic blueprint. Okay, so the DNA plus the protein going to give us the chromosome. Now you may have heard of chromosome because this is what's passed on from parent to child. And so a gene is the basic unit of heredity. So it's what determines, it is your genotype, and then there is the phenotype, which is the physical expression of your genes. Okay, so what are nucleotides? Okay, nucleotides are monomer units, so one unit, that are linked together to form nucleic acids. So when we take nucleotide plus nucleotide, we start to build nucleic acids. Nucleotides can be further broken down into a phosphoric acid, phosphate, plus a pentose sugar. So if you think back to carbs, we talked about sugars, and what's called a nitrogenous base. So it's going to have these three parts. So now if the pentose sugar is a ribose, then the nucleotide is more specifically referred to as a ribonucleotide. If the sugar, excuse me, and this is considered to be RNA. If the sugar is 2-deoxy, meaning it's not going to have one oxygen that the ribose has, deoxyribose, we call this a deoxyribonucleotide. Lots of words. And this nucleic acid becomes DNA. So that's the difference where the RNA and the DNA come from, is because of the pentose sugar and the different types. Okay. Otherwise, they both have a phosphate group, and they're both going to have nitrogenous bases. It's the sugar that's going to make them distinct. So the nitrogenous base, that's this third group right here, are found in nucleotides and are classified in two ways. We have the pyrimidines, I'm not good at this one, but pyrimidines and the purines. So a pyrimidine is a heterocyclic amine with 2 nitrogen atoms in a 6-membered ring, whereas a purine is a heterocyclic amine consisting of a pyrimidine ring fused to a 5-member ring with 2 nitrogen atoms. So I have images to show you what the difference between these is. So these are our pyrimidines. We have uracil, thymine, cytosine. And then the purine group has adenine and guanine. Okay. So we can see here that this is our 6-membered ring, and then this is a 6-membered ring plus the 5-membered ring. Okay. So the purine groups are a little bit larger than the pyrimidine rings. All right. So you're going to want to, I think sapling's going to have like 2 boxes like this, and it's going to tell you to sort them based on the structure, and you can do that because now you know that pyrimidine and purine, how they differ based on structure. These will be your 6 carbons, and this is going to be your 6 plus 5 carbon. All right. So you may have noticed up here that this said RNA only, and that that uracil is an RNA only, and thymines in DNA only, but that cytosine can be in both DNA and RNA. Okay. So if we look at the differences in the composition of DNA and RNA, RNA is going to have adenine and guanine for purine bases, so does DNA. But the pyrimidine bases in RNA are cytosine and uracil, where it's cytosine and thymine in DNA. And then DNA has that 2-deoxyribose, RNA has the ribose, but then they both have the H3PO4 in common. Okay, so this is a good table to know. Okay, multiple choice questions. I could see this easily being material that gets pulled out of this table. All right, so then we start building them, right? We've talked about the the bases, and now let's talk about actually building them. So in this case, we have the phosphate group, you have your sugar, and you have your base. Okay, and so you can see they kind of do look very similar once you get them next to each other, but the bases are different. Now I'm not going to ask you to draw these, okay, so don't worry. I just wanted you to see what the one nucleotide would look like, the single nucleotide, before we start linking them up. Now there are 2 really important adenine-containing nucleotides, and that is adenosine diphosphate and adenosine triphosphate. So it has your adenine base here and here. We have our sugar right here, our pentose, and then the only thing that differs on them is the number of phosphates. Okay, so you can see here there are 2 phosphorus, hence why it's called di. In this case, there are 3 phosphorus, which is why it's called tri. So ATP and ADP are part of our metabolic pathways. Okay, this is like the money in the cell. Okay, ATP is what's responsible for making chemical reactions happening, and that's through the breaking of these bonds. All right, nucleic acid structure. So a nucleic acid is a polymer made up of linking nucleotides together. Okay, so we're going to go back up here, and we're going to look at this first little formula. So nucleotide plus nucleotide. So we're going to take this entire group, and we're going to start linking them together with other nucleotides to make a polymer, which we're going to call a nucleic acid. The 2 that we're going to talk about are deoxyribonucleic acid, which is DNA. This is where your genetic information is stored, and then ribonucleic acid, RNA, which is the nucleic acid responsible for using the genetic information encoded in DNA. I don't know if you heard that loud slap, but my cat was eating my breakfast just now, and I just noticed it. All right, so when we start linking them up, this is kind of what it looks like, more or less. So this is the guanine, the thymine, the adenine, and the cytosine. And the DNA sequence, it tells you, is always read from the 5', and then we can't see down here, but there's a 3' down here. Okay, so the sequence, because it goes in this order, we're going to start seeing them spelled out something like this. Okay, so you can see here they're linking up through the phosphates and the OH group. That is right here. Okay. All right. So this would be considered more or less like the primary structure, just like the primary of the proteins. It's spelling out the amino acids. This is spelling out the bases. The secondary structure of the DNA is the double helix. If you've ever heard about Watson and Crick, you know that they're credited with the discovery of the double helix, which we saw kind of looks like the alpha helix, and that's the secondary structure. It's where the bases actually start to coil, the nucleoacid strands start to coil together to form an overall bigger structure. Now, there's something that made discovering the secondary structure of DNA harder, and that's there's a complementary base pattern. And this isn't like, I'm going to give you a complement. This is complementary, meaning you complement each other, like a pair, right? So you may have heard people dating, you say you complement each other, it means you fit together. You complement each other. And this is that there are specific base pairings in the DNA double helix that help offer that stabilization. Okay? So what does a double helix look like? Something like this. This is a computer-generated model of the DNA helix, so all of the little bubbles and different colors are representing different atoms. But then this right here is more or less a schematic representation. And the whole point of this one is to show us this complementary base pairing. Okay. So you have the sugar phosphate backbone, and then you have the bases which are being held together by hydrogen bonding. So while Watson and Crick, you can even see here, are given the credit for discovering this, Rosalind Franklin was a female crystallographer who was working at the exact same time, who was the first one who elucidated a picture of the structure. And with her X-ray structure, it's like taking a picture or like getting an X-ray of your bones, they were able to figure out the structure of the DNA. However, she passed away of cancer due to her exposure from X-rays, and was never given credit. Well, now we know. Now we give her credit, but she was not given credit at the time for her discovery. But now we have given her credit as far as her contribution to the discovery. All right, so complementary base pairing. Okay, so we're going to talk about the rules. So this tells you right here, the complementary bases engage in hydrogen bonding with each other. And thymine and adenine and cytosine and guanine are the two complementary base pairings. So if I'm looking at thymine right here and adenine right here, the complementary base pairing is happening between them like there. And then we have cytosine and guanine, which actually has 3 complementary base pairing structures. And this is really what they didn't quite understand, how they were matching up. They were trying to figure out the pattern. So the way I remember this, I don't remember if this was who taught me this trick, but I remember it always now. So the way I remember who pairs up with who is apple tree chewing gum, adenine and thymine, cytosine and guanine. Okay, now, but the problem is this is only true for DNA. Now, in RNA, there is no thymine, so the base pairing in RNA is we just swap out the T for the U, and there you go. So apple tree chewing gum. That is the complementary base pairing. So we need to remember that. Keep that in our back pocket for some questions that are coming up. So I'm not going to worry about the differences for now, okay? We used to talk about the differences between the different combinations that we could have and how using that was important in elucidating the structure. But I want to practice doing the questions, okay? So you need to get an in-class paper out. Today is the 20th for you guys, and send me in-class work. Okay, so I'll give you just a minute, and we're going to answer 2A through C, and then 3A through D, and 4A through D. So for this DNA segment, it looks very similar to the image we had before. It says identify the 5' and the 3' end of the molecule. The nice thing is that you're always going to start with the phosphate as being the 5', and then the end down here, the OH group that's free, is the 3' end. So it looks very similar to the image we saw before. So then it says, circle the atoms that comprise the backbone of the nucleic acid chain. So if we think back to our double helix, we said that the sugar phosphate was the backbone, okay? So we have to circle the sugar and the phosphates without circling the bases, because the bases are what's going to participate in our complementary pairing. So I did this with blue. And then it says, write the nucleotide sequence of this DNA segment. So in order to do this, we have to identify the nucleotides. So in this case, this is guanine, thymine, and cytosine. So you have to know, this is 2C, you have to know which of the bases are which. So if we go back up, you're going to need to know, where did it go? These. Okay. Make flashcards, but you want to be able to identify them. Okay. All right, so let's go back to our question. Okay, I'll leave that here. The nice thing about 3A, B, C, and D is that we're looking at DNA. And then 4A, B, C, and D, we're looking at RNA. And all you need to remember is the mnemonic I taught y'all, which is apple tree chewing gum. So if I have cytosine, it's going to pair with guanine in DNA. If I have adenine, it's going to pair with thymine in DNA. If I have guanine, it's going to pair with cytosine. And if I have thymine, it's going to pair with adenine in DNA. Now in RNA, all of it's going to be the exact same, except there is no RNA. There is no thymine. So cytosine is still going to pair with guanine. Adenine is going to pair with uracil. Guanine is going to pair with cytosine. But then thymine pairs with adenine. Okay. So knowing that base pairing the apple tree chewing gum will help you with this type of question. Okay, and I think Sapling's going to have you walk through a few of these to make sure that you can pair them up. Alright, so you should have 3 questions for your in-class work. And then we're going to talk about replication and expression, but just a few slides, and then I'm going to ask Dr. Morris if there's time, or Dr. Spencer, to show a YouTube video that I sent them that shows all this in action, because I remember learning about replication and expression, and seeing it on a slide where nothing's moving is really difficult, but seeing it in action for some reason made it more clear to me about the moving parts. So we're going to talk about it first, and then you'll watch a video. So replication and transcription are really important because it's how we replicate and express genetic information. So replication is the process in which DNA in a dividing cell is copied. So we're making a copy of our DNA. Transcription is the process in which RNA is synthesized from a DNA template. Now, the thing that we haven't talked about yet is that our RNA goes on to make proteins, which your body needs. Okay, so this is called transcription, and then this process is going to be called translation. Okay, so often this is called the central dogma of biology. Okay. So it's a very complicated process. Okay. One of the first things that has to happen in the DNA replication is that there has to be what's called an unzipping process, because the DNA is zipped up in a double helix. And then during that process, you have proteins that are going to come in, and they are going to be responsible for helping to move along and replicate. So you see we have the DNA polymerase, which is unzipping. It's an enzyme. Unzipping and zipping. And then you have these right here, which are making copies. Okay. So it's very complicated. The thing is, your body is doing this all the time. In fact, it's doing it right now. And the other thing is, you don't have to think about it. You don't have to tell your body, okay, make a DNA copy, or transcribe this to RNA, or translate that to a protein. And it's doing it very quickly. And there are going to be examples where you have mutations, and this will result in illnesses and things like that we'll see. So after that, you have RNA that's going to be, so we made our copy of our DNA copied. Then we're going to take our copy of the DNA and make an RNA. Okay, so now we see down here there's uracil instead of thymine. So it's going to, the uracil is replaced by the thymine in the adenine base pairing. Okay. But we're going to see this on a video. So in this, there are three types of mRNA. There are three, there's mRNA, rRNA, and tRNA. And the reason why I want you to see these is because in the video, these three RNA types are going to have different jobs. Okay, so they have different jobs in this whole process. Okay, so the mRNA is what's coding for the proteins. The rRNA, which stands, so it's messenger RNA, ribosome RNA, which is a component of ribosomes, and T stands for transfer RNA, which is the adapter molecule that brings the amino acid to the ribosome. Okay, so we're going to see that. Now, what is a ribosome? The ribosome is a cellular substructure where proteins are synthesized. So this is where RNA becomes proteins, which was that last step. And with that, we're going to have two different types of things, which is a codon and an anticodon. And a codon is a set of 3 nucleotides on the mRNA that specifies a particular amino acid. So you'll have 3 nucleotides that equals 1 amino acid, which is what the building block for a protein is. So this is how we go from the nucleotides in RNA to the amino acids to form the proteins, because there's not a direct translation. So we have to transcribe them from one language to another. And then we have the anticodon, which is a set of 3 nucleotides on the transfer RNA that is complementary to and pairs with the codon on the mRNA. So this is kind of the process that really is interesting to see on the video that you're going to watch. So what does the tRNA look like? Looks like a T, which I always loved. Okay, so here's the tRNA. It possesses the anticodon down here, and then it's going to match up with the codon. Okay, so the codon and the anticodon will match up, and this is going to be on the mRNA. Okay, so I'm going to go ahead. I think it's probably been, I didn't start my timer on my phone right away, but it's probably at least been 25, maybe 30 minutes. So you're going to watch this video that's not very long, and then we're going to pick up with the protein synthesis and genetic code and mutation and viruses next time. I hope that all is going well. Please email me if you have any questions or concerns, and I'll get back to you as quickly as I can.